Welcome back to ciLiving. I'm your host, Jaclyn Friedlander. Science meets society at the University of Illinois. They offer a variety of programs and events to drive positive change to the community and the wider world. Joining me now is Daniel Urban. Thank you so much for being here.
Thanks for having me.
All right, so what are we doing today?
Today we are going to be extracting DNA. So I'm from the Institute for Genomic Biology. And genomics is a big scary word that most people shouldn't be afraid of, because genomics is just the study of the genome. The genome is all the DNA in a living thing. Everything that's alive has DNA, so we're studying it all today.
Okay.
Everyone's probably seen something typical like that double helix structure representing DNA, and here we have it in a 3D printed model. Here we have it in a little origami model.
Right.
You can even have a DNA plushie.
Plushie, that is so cool. And so what are the different, what is this actually representing, right? So we've all heard the word DNA, we've all seen these double helixes, but what is it?
DNA is typically referred to as the building blocks of life. So, right, all these little, I don't know if you can catch it on there, but there's little letters on there, A's matching with T's and C's matching with G's. These are the base pairs. Adenine matches with thymine and cytosine with guanine. Those base pairs make us who we are. They're the blueprint of us. So a human being, for example, we have our entire genome, our full set of DNA, has 3.2 billion base pairs in it.
Oh, wow. Okay.
And we all think of that, it's microscopic, of course, it's inside every single one of our cells.
Right.
But it's actually 3.2 billion is a lot, right? So you think you should be able to see that. It shouldn't be that tiny, and you actually can. It's just that it's wound up really tight and it's compacted into every one of those cells. But if you actually break it out of the cell and open it up, if you were to unwind 3.2 billion base pairs, and from the floor up, it would be as tall as me.
Oh, wow. Okay. And so we're going to today pull that out of these strawberries?
We're going to pull that out of the strawberries. It's
okay.
a simple experiment. So like I said, it's microscopic, but if you open it up, you can see it with your naked eyes. And I think that makes it a little bit cooler. And this is a simple experiment that you can do at home using simple ingredients.
What do you need?
So you need to make a lysis buffer. And that's actually a pretty simple thing to do. That buffer just contains water, dish soap, and salt. That's it.
Okay.
And then to open it up a little bit further, you need a little bit of alcohol. So we're using rubbing alcohol for that.
Okay.
And you can do this with a lot of different fruits at home. We're using strawberries today. It's a very typical one that people try this with because there's different quantities of DNA in different organisms. So
I see.
A lot of things are considered diploid. It means every cell has 2 copies of your DNA. But strawberries are a little bit special. They're actually octoploid. So every single cell has 8 copies of their DNA. So it almost means it's really hard not to get DNA out of a strawberry because there's so much of it.
Right. Well, we have about 2.5 minutes.
All right,
so how do we do this?
We're going to do this real quick. So I'm going to give you a strawberry, and if you can open up your little plastic bag with the paper towel in it.
Perfect.
Stick it in between the paper towel right there.
Keep it in the, got it. Yep. All right, stick it in
So you see I've made mine in the little pouch in the paper towel.
Got it.
We're just going to close that up, and we are going to smush it.
Smush the strawberry.
We're going to smush this strawberry, and we're using the paper towel as a filter, basically. We're trying to keep most of the pulp in there.
Right.
But mush all this juice up.
Okay.
And now we're going to take this little lysis buffer that we made.
All right.
And you open your paper towel just a little bit, and I'm going to squirt some of that in. See?
And so what is that mixture?
This is water, soap, and salt.
And salt
the
all mixed
soap
together.
helps break open the cell.
Okay.
And then the salt helps separate out all the little other cellular components and pull them away from the DNA to isolate it.
I see.
And then once you have that done, you should start seeing a little bit of bubbles in there.
Oh, yeah.
Mix it around because,
from the soap.
Mm-hmm. Now, if we can get a little bit of the liquid out, try not to get the pulp.
Okay.
Into like one of your corners.
Perfect.
I think you've got it pretty good there. What I'm going to do is I'm going to nip off your corner.
All right.
Take that.
Cut it off.
And then if you can squeeze some of that liquid into this tube where we already have some of that alcohol in place.
Okay, so the little tube just has the alcohol in it.
Yep.
Okay.
Just that. I'm going to put a little more in there.
Do I need more from the,
if you could.
From the bag? Got to make sure we get a good sample size,
I want to make sure we get enough in there.
Okay, perfect.
Awesome. All right. I'm going to cover that up. And can you just shake that for me?
Shake it. You got it.
Up and down. Let's see what we got here. And survey says.
Oh, that's so cool. Oh, my goodness. Yeah, you can actually see the DNA in there floating around.
And so DNA is usually water soluble, but it is not soluble in alcohol, so it precipitates out of it and becomes visible to the naked eye right there.
And how many, I mean, a little sample that size, how much DNA is in this little vial?
There's probably several strands of that in there. DNA for strawberry is less so than for humans, so there's about 210 million base pairs in that. So you probably have several strands of it. And one of the simple little things we like to do is you can just pop that on a little string and make a DNA necklace out of that.
Make a strawberry DNA necklace. That is so cute. Thank you so much for teaching us how to do that. What a simple thing that you can try at home. That is so cool.
And you can do it with all kinds of other fruits. I've done it with kiwis, with grapes, with bananas, with even something as hard as a pumpkin. I don't recommend. You really have to mush hard for that one.
Right. You really have to work for it if it's a pumpkin. Well, thank you so much for being here, Daniel. I really appreciate it.
Thanks for having me.